also this morning we have two external invited speakers, and so I really want to encourage people to tap into their expertise and for lunch as well. But the first is Professor Ji-Xin Cheng from Purdue University, who has joint appointments in biomedical engineering there as well as chemistry, and he I guess some of the recognitions that he's received over the years. One includes a Purdue University faculty scholar, which I believe is kind of a campus scholarship program for outstanding faculty. And then also, I noticed from his CV, he was an outstanding young scientist award from the Chinese National Academy of Sciences. And but he's been working on using and developing new optical imaging techniques, notably CARS and other spectroscopy techniques, to be able to look at the nervous system, the neural tissue, and many other different applications. So I think he's going to present a wealth of different ideas and topics today.
Thank you. Thank you, Steve. So I changed the title a little bit from nervous system to life. So I try to give you a global overview of this using chemical bond as a signal to see different systems in so why it doesn't work. It's on. Oh, it's on. Is it working? Is this the right one? I can use. I have another one, my own stuff. It works. I have another one. So. This one should work. Okay. Okay, this one. Thank you. Well, thank you. So let's start with this picture. So this is a book I bought for my 4-year-old son. So, so here the job is where's Wally. It took us 10 minutes to find. Wally is here. It's a small guy with a hat. So this presents a big challenge when we study a biology system. Right? It's so complicated. You have so many features, so many structures, and it's even dynamic. Here is not dynamic. But the key is that we eventually find a Wally. So why we can find a Wally? Because Wally has a special pattern, and our brain just did a pattern recognition to find a Wally. So this presents a challenge and a hope to study a very complex system. However, we don't do this in real life. So this is what we do. We digest the whole cell and pick up and centrifuge and do in vitro biochemical assay. So this has been used every day in our biochemistry labs. So this method tells the presence of the molecule, but they don't tell us where the molecules, the dynamics, and how the molecules work together, which is the foundation of life and disease. Right? But there's another way. We want to visualize Wally, right? And how to do it? We label the molecule. This is also a very powerful tool, especially with this super resolution, that structural illumination or single molecule detection. We now can visualize details of molecule. Meanwhile, that we have to label the molecule, so we have to know the molecule in order to see the molecule. So we cannot study the composition of an organelle. And also, in many cases, the labeling may perturb the structure of a compartment, such as a membrane. Right? You don't have too many. The membrane is very thin. We cannot put too many molecules there. So these are the ways we use, but it is not the way that a child finds a Wally. So we want to go back to do science as we do in normal way. So the thing is that can we see the molecule in the intrinsic property of molecules of the system, and that is called label-free imaging. And there are many ways to do label-free image, right? So I will present one of them, which I believe is the most informative. The reason is because that every molecule has this chemical bond vibration and produce a fingerprint vibration. So each molecule is different. So that's why it contains lots of information. So I have been studying this for actually 20 years. Since I was a PhD, I work on this gas phase molecule, studying locomotor vibration, which means that all the chemical energy focus on one chemical bond. So this tells us that through the spectral analysis, you can see how a molecule vibrates. So this is in gas phase by FTIR. This cannot be used to study living cells because IR absorption, water has a strong IR absorption. Instead, we could use another vibration technology, which is called Raman scattering. So Raman scattering from water is very weak, so we can image the biomolecules. And as you can see here, these different protein, DNA, RNA, lipid, they have a different fingerprint, right? That's the hint. And also Raman spectroscopy has been a very powerful tool as a sensor. So this is using a tweezer and they do single cell Raman sensing of a single cell status. However, cells have so many molecules, right? So many different molecules. It's impossible to resolve where the signal comes from if you take a Raman spectrum from a whole cell. So that presents a big challenge, and how to solve that problem. Well, the hope is here. The hope is because the cell is not a bag of molecules. So when we do the omics, when we do this single cell Raman, we essentially treat a cell as a whole bag of molecules. Right? But the cell is actually structurally organized and is highly dynamic. So the hypothesis is that if we can take a spectrum very fast at every pixel, then the spectrum is simplified. Because if you look at a very small volume, you don't have so many molecules. You only have a few molecules actually. And then you can study the status of a membrane, of a mitochondria, of a nucleus based on this spectroscopy profile. So we call this a vibrational spectral imaging of a living system. Well, this is a concept, right? But we face a lot of challenges because conventionally that a Raman take 1 second or a few seconds, and that means it will take a few days to record an image based on, and that's not possible to capture dynamics. So I have to mention that about 10 years ago, there's a technique called CARS, which is pioneered by Sonny Shi and others. And I had the opportunity working with him as a postdoc, developing this. One of the so the CARS actually involved these two beams, and they produce signal at the anti-Stokes frequency. So in general, this process is coherent, so it adds up. You have 10 molecules in signal increase 100 times. So that's the reason that the main reason that CARS produce a larger signal than spontaneous Raman. So that time we actually never dreamed that we could take a Raman spectrum as fast as fluorescence. Instead, we use a compromise approach. We focus all the energy on the single Raman band. So this is a single color excitation. And this produces a very big signal at the anti-Stokes frequency. Meanwhile, this process, actually there are other processes happen at the same time. It's called stimulus Raman scattering. And this is featured by a gain at the Stokes beam and a pump and the loss at the pump beam. So this pump Stokes means the frequency of the beam. The energy difference is resonant with a molecule vibration. So this energy transfer actually go to the molecule and excite molecule from ground state to excite state. And CARS is virtually a parametric process. And this all happens at the same time. So recently that the same configuration has been used to do this SIs, which you can do SIs and CARS on the same microscope. So this is what you do. You use two narrowband beam.
Can you read the slide and explain again how these cars were on base of signal generated?
Oh, that's it appeared a new frequency. So it's virtually a parametric process. So it's a four-wheel mixing process. And this four-wheel mixing process signal can be enhanced when you have a Raman resonance.
So the excitation, the frequency of resonance changes, like what do you still shine something on the molecule? So I don't know the basics of Raman. So okay, explain briefly like how this one is so strong and works.
But as I mentioned, that this process essentially the energy transfer here is because there is energy transfer from the field to the molecule, and this is by the Stimulus-Raman process. The cars is appeared. The cars essentially does not involve energy transfer, so it's a parametric four-wheel mixing process, and that can it's hard to explain by. But we can talk more. Yeah. So yeah. Okay, because this is quite there's a whole theory describing this process. So technically, how you do this process is you combine the two beams into one beam and send it into a sample. And this, for cars, you can use a PMT to measure the car signal because it appeared a new frequency. But the Stimulus-Raman process is actually appear the same frequency of the laser. And to do that, you have to modulate one of the beam. And this Stimulus-Raman gain produces a very tiny modulation on the other beam, the Stokes beam. And this very small modulation can be picked up by the lock-in amplifier. Since you know the modulation frequency, and you tune the lock-in to pick up this very small SI signal. So what's the difference between cars and SIs? SIs, in cars, there is a four-wheel mixing, which is essentially a non-resonant background. And the SIs is free of this non-resonant background. So talk about neuroscience, that one of the things we did at Purdue, the first project is to using cars to bring cars to neuroscience by imaging these myelin sheaths. The myelin sheath is actually a very important structure that the surrounding axon is very important for impulse conduction. And myelin give a very strong signal from the CH bond. So this allows to study the myelin degradation and regeneration in real time. So I give you one example that this is a parallel myelin retraction in real time, and you can visualize this. So this happens actually upon a high frequency stimulation, which is a therapy. And this is gained because we have very high speed, that this is 1 frame per second. So you can see this myelin dynamics in real time. So this is gained by the high speed of cars microscopy. The other application is used to study obesity because these lipid droplets give a very strong signal, so we can visualize this in a few groups using cars or SIs to see these droplets in C. elegans. Or we can study how the lipid enter the body through the small intestine. But here then we have a question, right? Because we focus all the energy on the CH bond. There are two difficulties here. One is that we don't know what's inside these droplets. We don't have the spectral information. Second is that all the molecules have CH bond. So even when we see some features, we actually don't know whether they contribute by lipid or other molecules. So we need to gain the spectral information back. And the simplest way to do this is you have two narrow bands, and if we scan one beam, then we can record a stack of images. So we call this frame by frame hyperspectral imaging. Well, this can be done with the existing picosecond setup. You just scan the laser one by one. Another thing that we realize that we try to correct the mistake we made ourselves is that to move from CH to fingerprint region. So initially we thought, and it's a common thought, is that fingerprint is so complicated. Right? Every molecule has a fingerprint. So there's no way to resolve the species of molecules. So then we always take a compromise approach. We go to CH bond. But the thing is that the CH gives you a huge signal, but every molecule contributes to CH stretching vibration. So it's actually the results can be easily misleading. And I will give you an example in a few minutes. If you go to fingerprint, each molecule is different. And as I said, if you can record a fingerprint spectrum from a very small exciting volume, the spectrum is simplified, and you actually can resolve the species. So now this involves a thing that how to analyze the data. If we have so much data, now at each pixel we have a spectrum. So we have a XY lambda 3D matrix here. There are many ways to do this. So here we use the one way that is called a multivariate curve analysis resolution. The beauty of this method is you can actually analyze a relatively unknown sample. So what you do is that you decompose into 2D matrix. Then you can fit this with the initial estimation from the principal component analysis to two matrix. One is a concentration matrix and the other spectral matrix. You can then refold this into a concentration map of each component, the major components. So this is a real chemical imaging. Yeah, please.
The ALS, is that alternating least?
That's right, yes, yes. The constraints is very interesting. Yeah, the constraints is that so if you do least square fitting, the constraint is 100%. You fix. So that means that you import a reference, and then, but then so the constraints you can add. So this one is that you can, this method beauty of it, it can be very soft. Means that you can release the profile. So you made the initial estimation, but the fitted profile can be different because you don't know the real profile in the tissue, right? So that means, yeah.
Do you have any prior assumptions on the spectral profiles at all?
I'm sorry?
Do you have any prior assumptions on like the spectral profiles?
You don't have to. I will give you an example. Yeah. Okay. So I think that's the thing. This is a thing that also the challenge and the opportunity of this method, right? So because of this, we now have the power of discovery, which is not there in fluorescence microscopy. So this is a simple example we have published. That here the goal is what's the goal? The goal is that the volume now is cholesterol. I want to know where is the cholesterol and in what format in the tissue. So this is a artery tissue. And how to do it? Well, you need to find the pattern, right? Because it's a pattern recognition. And the pattern we found is that the cholesterol has a sterol bond. And this sterol bond is different from other CC bond. The reason we use CC bond is that because CC bond is a strong Raman band, give a strong Raman signal because of the pi electron. And then, so these peaks are close, but they are not exactly the same. So this gives us an opportunity. If we can record a spectrum, then we can use this pattern to recognize the cholesterol crystal. And also we actually can determine the amount of cholesterol in each droplet. So this is in cardiovascular disease, there are a lot of cholesterol in the artery. And also we can recognize the protein by the amino-1 band I didn't show here. So that's one of the example here.
So we could show arteries that are compromised in some ways, like the so we get a different signature if there is some degradation of the artery.
Yes, that's right. That's right. Yes. But now I want to give you another example. That this is another example. Now it's still under review. That I could recognize an unknown feature. So this is a history because C. elegans and studying lipid metabolism in C. elegans is it's important to understand the relation between imaging, between obesity and aging process. So previously we and others using a CH1 imaging the droplets. So we don't know, right? So all the molecules contribute to CH vibration, and that was misleading. Now we found that by fingerprint imaging, we can record many species, and these species, including these liposome-related organelles and fat droplets, the oxide lipid and protein. So these are the 4 major species, and we can quantify the difference. But this is between this DEF2, which is the insulin signaling pathway. So how we do this? Let me show you how we do this. So we first pick a stack of 100 frames in this window. Well, this window is determined by the spectral width of the femtosecond pulse. So we can broaden if you have a better laser source. And then you see a lot of organelles, and you can determine the main component by principal component analysis. But seeing that these organelles, if we pinpoint, you can pinpoint this in real time using a software we have developed. And they have different features. And these spectrum are slightly different. So we don't know what's inside, but this can be an initial guess, right? So we can pinpoint. And now we largely identify 4 features. So 3 are from the intracranial organelle, and 1 is from this layer of the membrane, the skin. So if we use this as an input, now you can do multivariate analysis. And this produces a very beautiful picture for us. So now we see that there are 4 major components. And now we can go back to the literature because the Raman spectrum for every molecule is known. Right? So now we can go back to study what's inside. And this takes a lot of work. I want to show you the result is that we found, for example, that the oxide lipid, which is the purple one, and these are many dots. And these are featured by this peak at 1680 for aldehyde. And then the fat droplets can be featured by 1655 for CC double bond and the ester bond. And the most important feature is lysosome-related organelles. And we found a very strong peak from cholesterol, which is at this 1669. And the protein by the amino-1 band. Of course, to confirm this, we have done a lot of study in this paper that using glutens, that if you deplete the one chemical, and we found that this peak will disappear, and I will also that you can. So this is another evidence for the oxide lipid that has another peak at 1550, and that's from retinol. And these 2 together actually identify a species called retinol aldehyde, which is aging product. So this shows the distribution of lipid oxidation. So this study showed for the first time that the cholesterol is stored in the lysosome-related organelles, and nobody knows this before. So this kind of show you a capability of this method is that we can identify the unknown composition of organelle, and this is not reachable by a labeling method.
Qualified concentration.
Yes, actually, these all map concentration actually, and of course, if we using a reference, then we can get the real concentration. So this concentration pretty high is minimal, pretty high. Local concentration are minimal. Well, this is show you that a transition from in vitro spectroscopy to spectral imaging. But the transition from in vitro to in vivo spectral imaging is more difficult. Because in the example I show you that it still takes a few minutes to record a stack of images. So we actually have to using a laser to slow down movement of the worm in order to take the static image. So I mentioned spectral imaging is not simply spectroscopy plus image. We need to overcome challenges. So today I want to show you two things. One thing that we have asked is that we ask one question: can we require spectrum as fast as you require fluorescence, like in a few microseconds? Because if that can be done, we can study living system, and we can study dynamics, how it change with the memory change with the cellular process. The second question we ask is, can we take a spectrum at a few centimeters inside the body? If that can be done, we can do spectroscopy CT, so that we can do diagnosis with information. So one thing to do this is to do a broadband excitation. So if you use one narrowband, the other broadband, then you can do broadband CARS or broadband SI. So I have done broadband CARS many years ago as a postdoc, and we stopped. The broadband CARS has been very useful by a few groups, and it reached a speed of 30 milliseconds per pixel, and that's much faster than Raman, but still not fast enough to capture a dynamic process. For example, we know that in about neuroscience, we know that the action potential takes 1 millisecond. And to capture that, you need a speed of less than 100 microseconds to capture that process, right? So the method we use is to do a broadband stimulus Raman scan. And that's the idea, is that you use a narrowband and a broadband. So in this case, the Raman spectrum is coded in the broadband laser pulse. You measure the whole laser pulse, and the laser is used as local oscillator. So in technology, this is the reason that we gain the speed, because the local oscillator boosts the signal. So we got lots of photons, right? In CARS, we don't have enough photons. That's the difficulty. But here the difficulty is that how we extract the signal from the laser beam. So everything we measure the laser, so that's you can do this experiment at ambient light. So that's a beauty of this. So here we have quite a few methods. So I will present one that we have finished, and this is through this using a circuit to extract the signal from this. And this circuit is from this model of a cochlear model. So this is a class I teach and produce. So the ear we can hear different frequency sound is because it's a stage of our LC circuits. It amplifies a specific frequency of the sound. So this is good because in SIs we actually know the frequency, the modulation frequency of signal. So we essentially can use this to amplify all the signal specifically. So this works pretty well. So this is what we built. So you have a photodiode measure laser, and you can use a circuit, which this is a concept. The real circuit is more complicated, but this can amplify the SI signal without locking. So this is a different method, different way to do it. And you can use a rectifier. You can buy this rectifier with maybe $100, and then you can replace the whole lock-in amplifier. So why this works? Lock-in was considered to be the most sensitive instrument in physics to measure heterodyne signal, because the bandwidth of a lock-in can be as narrow as 0.1 hertz, and there's no way we can do that with a circuit. The reason it works is that now is high-speed imaging, so the pixel dwell time is only 10 microseconds, and that corresponds to a bandwidth of about 100 kilohertz, and that is easy reach, can be realized by this circuit. So this circuit exactly match the pixel dwell time. So that's how it works. So initially we are happy because this will make things much cheaper. But the real value is that this circuit is so small. This is made, so we can easily make an array, a sample array, and this allows us to record a spectrum. Then this power increase, right? Because you now can record a spectrum at the same speed as we record a single color previously. So initially we now can record a 30,000 Raman spectrum per second. So that means it's 30 microseconds per pixel, right? So this is how we do it. We actually we have 2 femtosecond beam, and one is we actually learned a method from Chris Xu with here that to spectral compression from femto to pico, and the other one is that they use a power shaper and send this to array here so this array amplifies the stimulated Raman signal at each color. So this allows to gain a spectrum. So this is how we do it. So this circuit is nice. It input DC and AC. So DC is virtually the laser beam, and the AC is your signal. The AC over DC is exactly Raman. So it's actually perfect. And this is exactly match with the spontaneous Raman. The difference is that this spontaneous Raman take 1 second, and we can take 30 microseconds. So this can be very useful. For example, this is an example that you can do in vivo imaging of drug diffusion. So previously people have done this, but they have used a CD bond and modify the drug with a CD bond to see an isolate Raman band. Now we don't have to. We can use any natural molecule, and because what we do now is pattern recognition. It's not an intensity image anymore. So as long as the pattern is different, we can separate them in different channels. And this is a speed happens that they change in seconds. So we are fast enough because we can do microseconds, right? So each image takes 1 or 2 seconds, so we can see the diffusion dynamics. But the most important thing is that we can now do flow cytometry. The flow is very fast. Right? You probably want to measure 1 million cells per second. That means that you need 1 second. But so this example, this speed flow very, very fast. And we do night scan, we can capture these speeds. And we can separate them easily. That is because at each pixel we have a spectrum, and I zoom in this place, and the spectrum is different. So you can easily separate these 2 beams. It looks similar, but in scattering, in really scattering, but now they are very different. So this opens the opportunity to do a spectroscopic flow cytometry, label-free, with many channels. We can now do 16, but we can do 32. So this is an example that this is a flowing tumor cells, and you still can map the chemical content of a fast flowing tumor cells. So in other words, that if we have 10 microseconds with a better card, which means we can analyze 100,000. We can analyze 100,000 cells in 1 second with spectroscopy in a label-free manner. And this can allow high throughput single cell analysis. Well, another thing I want to tell you is that how to better sensitivity. So this allows high speed, right? But the sensitivity is still limited because the Raman cross section is small. But this is another direction that if you can make a molecule that is the Raman signal very big, then we can lower the concentration. So we have studied for 2 years, and we made a series of probes. The most useful one here is that with 2 triple bonds plus a phenol ring. So this structure exactly mimics the structure of cholesterol. And the triple bond is unnatural bond. It gives a signal that different from endogenous molecules at 2100 wavelengths. But this was not made by me, it's by my colleague, Ji-Xin Cheng, who is an organic chemist. So here I show you the signal. So we compare the different species, and we found that the signal from the CC triple bond is actually from this molecule is actually 122 times bigger than the CO bond from solvent. But solvent is 100 mole, and this is 15 millimole. That's why you see they are comparable. Well, other people have published and they use triple bond as a contrast agent. But actually, in reality, that the triple bond is toxic, and the toxicity is about 20 millimole, 20 micromole. But the sensitivity is 200 micromole. So virtually, it's difficult directly using triple bond alone to study biology. Maybe we can prove the concept, right? But we cannot follow the dynamic process. Well, the IP here is that we found accidentally that by add a ring to the end, that make it non-toxic because the ring is inert. But that also increases the signal by another 10 times. So this can be quite useful. So now, for example, we can study a biochemical process of conversion of phenoldiene cholesterol to cholesterol ester. And this is by an enzyme called ACAT1, right? To study this cancer metabolism. So if we treat the CHO cells, actually this will go to the lipid droplets, and if we tune away, it's gone. That this is CH imaging. And if you inhibitor, actually they are no longer colocalized with the droplets. So which means that we actually can study this biochemical reaction. So the one stored in the droplets is no longer the cholesterol, it's actually cholesterol ester. And this is a very important process of cholesterol metabolism inside cells. This is important because the current gold standard is cholesterol body P. That's the best one you can find in the market. And the body P is very hydrophobic. So body P simply pour cholesterol to the place the body P want to go. So in other words, that in the mature cells, it go to the droplets. But if we use the inhibitor to block this biochemical process, the image has no difference with the control. So in other words, that they go to droplets directly without any biochemical reaction. So that shows, I believe this is one advantage that the Raman can provide. Because the Raman secret from chemical bond, we can design some structure that does not perturb the physiology of molecules, especially small molecules. For bigger ones, we probably don't have a chance because proteins, right, those are very powerful, the fluorescence. So one example that this can be used to study there's a disease called the Niemann-Pick disease. There's a cholesterol storage disease. So currently the gold standard, you have to fix a cell and do the biochemical assay or using a probe called you have to fix the cell and do this. And now we can do a living cells before and after treatment without the so you can study dynamics, and then after this drug, we can see that there is cholesterol mobilization. So this is very important because this can do high throughput imaging evaluation of this cholesterol lowering drugs. Well, there's another concept I want to tell you is that this structure, this yin and yang structure, I put it on the cover of a recent review article here. So a big problem of this community is that when people work on cars, SRS, and they try to see that, well, we can beat Raman. And we realize that that's not true because you and your opponent always complement each other. The same for a technique. Coherent Raman and spontaneous Raman complement each other. So this picture tells you exactly my thinking is that the coherent Raman cars SI provides the speed and the spatial resolution. The spatial resolution because nonlinear provides 3D spatial resolution. And this was achieved by focusing the energy on a single band or a narrow window. Right? So, and the spontaneous Raman. So in this yin and yang picture, this means the weeklies. The weeklies spontaneous Raman is very slow And the strength is here. The strength provides the bandwidth, it provides the whole bandwidth. So people in MIT, they can detect the trace of glucose. When I was there in February, they told me they can detect the trace of glucose in the skin. And they achieved that simply because they fully utilize the bandwidth of spontaneous so that they can separate the change of glucose concentration from the skin tissue. But again, that was achieved by a very long time of data acquisition from the skin tissue. So now, if we put this together, we can gain speed, spatial resolution, and bandwidth if we put this together, right? And that's seems like a no-brainer idea, but it's very useful. So now we can do high-speed SIS pump probe or CARS imaging. And because we use a picosecond beam, we can do a picosecond Raman on the same microscope. So in other words, if you see an organelle by CARS, SIS, and you can use Raman to determine the composition with the best bandwidth we can have. Well, we cannot do better than spontaneous Raman. And so this leads to a very interesting discovery made recently and published 2 months ago, is we found that this cholesterol ester as a metabolic marker of cancer aggressiveness. So what we did is we actually scanned every stage of prostate cancer from normal to metastasis of human samples. Again, that's a beauty of label-free, right? So we can study human because we cannot use GIP to label the human specimen. And you see these very tiny droplets, a beautiful image in SIS. So that tells you the droplets. So initially we thought that's fat, obesity stuff. But then when we do the Raman spectroscopy, we actually found that this is actually the structure for a molecular cholesterol ester, and this is featured by these very tiny bands that are very matched with literature. So it's very difficult using CARS or SIS to find all these very small features. But Raman can do it, right? These small features all reproduced by spontaneous Raman. Of course, we've done a lot of biology to confirm this molecule, including mass spectrometry, proteomics. So this allows us to continue to study biology. So I want to tell you the story behind this. So this is that the prostate cancer is controlled by this androgen receptor. So this pathway was discovered in Chicago in 1930s, and that led to a Nobel Prize in 1960s for treatment of this androgen therapy. But then the disease always came back after 1 or 2 years, and the disease became androgen independent. And people propose there's another pathway that is with the PI3K-AKT pathway. It's a metabolic pathway. So what we found is that this cholesterol accumulation is driven by this P10 loss. P10 is a tumor suppressor gene, and the PI3K-AKT, and there is a SHREP protein which controls the lipid synthesis. So what we found is that this actually enhances the lipid uptake from LDL receptor and the LDL uptake from blood. And this cholesterol is converted to the cholesterol ester by this ACAT1 enzyme. So we could, if we block this step, then we found that the cancer actually proliferation is much reduced in vitro, and also this data of the cell cycle progression stopped and induced apoptosis, and also that actually the more important is that this reduced the cancer migration and invasion, and that's not a surprise because cholesterol is a very important molecule in the membrane, so it changes the whole membrane. And we also have in vivo data show that the tumor proliferation much reduced, and this is not toxic because this process is very cancer specific. And we found now same phenomenon in pancreatic, lung, colon, almost every cancer. So what happens if we block here, we actually downregulate the whole pathway. The reason is because free cholesterol is a substrate for the SHRB, so we can downregulate the SHRB, and also we found that we can reduce the cholesterol uptake by 10 times by downregulation of LDL receptor. And this also reduces the uptake of these polyunsaturated fatty acids, which is also a feature for cancer growth. So the significance of this is that after the hormone therapy, then the disease becomes hormone independent. At that stage, these drugs are non-specific. So this is further reducing the activity of androgen receptor, but they only extend the patient lifetime by 2 months. So we wish now that with our new pathway, if we can extend the patient life by another 1 or 2 years, that will be bring a lot of benefit to the patient. So I would briefly, do I have time?
Maybe, yeah, 5 minutes.
Oh, 5 minutes. I will use 5 minutes to summarize a new direction that we have, that I mentioned that this field do have a problem. That the second question I have is that the imaging depth is very limited because we use biophotons, and this is very good for high resolution imaging, but we cannot go deeper. So a few years ago, we tried to study this plaque, and people always ask me, can you do it in vivo? And that's not possible because we actually cut the cross section, and that cannot be done in living patient. So that triggered us to build a new method called the photoacoustic imaging chemical bond vibration. So we spent 2 years to build a photoacoustic SIs. So that time SIs was not published. But we didn't get any signal. But accidentally we found a signal that is from overtone excitation. And so why photoacoustic? Because the ultrasound, the penetration into tissue is much easier than photons. And second, by measuring the phase of the sound, we can tell the location. So we can do tomography by ultrasound. So this tells us not only the depth, but the whole big volume. And this depth and the volume are 2 essential parameters for diagnosis, because the doctor wants to see a big volume to make a decision. So this involves absorption and pressure wave generation and acoustic detection. So previously, Li Hong Wang and others have used this to measure this blood absorption as a contrast mechanism. And we now show there is another contrast mechanism using overtone excitation. And to do that, by doing that, we actually can measure the tissue content beyond the blood. And that's essential information for disease diagnosis. So this shows you the physics here that this by this Boson distribution. So when the relaxation of vibration to the ground state, and this produces heat, and the ultrasound if we use pulsed generation. This gives you one example that so if you go to visible, you have this blood absorption and but the second overtone appeared at 1200 nanometer, and that gives you a signal that is not overlapped with the water absorption with blood absorption. So this is a place that we can use to do image of the other molecules. And the first overtone appeared at 1700 wave number. So one thing you can do is that you can image, now we can image a plaque directly from the lumen side. And with this endoscopy, we can achieve 3 millimeter. So we can have a resolution of 100 micron. So that'll be good enough to do this. So this is our latest stage. And now we have built a catheter, so-called intravascular photoacoustic catheter. And we have built our own laser called Raman laser that with 2 kilohertz. So this allows, we have reached a speed of 4 frames per second. And the beauty of this is that this can be coupled with intravascular ultrasound. So this is a medical practice. but it gives the structure information. And now we can give chemical information on lipid, and you can overlay them, and you can see why these are not complementary to each other. But this lipid information is very important for doctor to judge whether we should put a stent or not in the artery. So this shows you the current tools, and they still have difficulty to judge whether plaque is vulnerable or not. So we believe that the intravascular photoacoustic can provide this key information of lipid core size to do two things. One is to study the vulnerability of plaque, and second, to evaluation of the cholesterol-lowering drugs in patient. So this is a product that we are making, and we have a business now to make this product that is in the intravascular, but we have dual modality to do ultrasound and the photoacoustic imaging sequentially. Well, this is our dream, that we actually eventually want to put the detector here and see the artery, right? Because this will be completely non-invasive that the doctor can use every day. So if we can put it in the neck, we can check the carotid artery, or we can do this. So this will be like spectroscopy CT. But this lead us to increase the depth to a few centimeters. Right? Millimeter is not enough. So is that doable or not? So I will show you some initial effort. So the thing is that this is possible if you diffuse the photon first. So you excite the whole organ, and now you can put more photons to the body. So this is called tomography. So you can rotate this transducer, and that we use this proof the concept. We need a laser, so we have built a Raman laser with high energy, like tens of millijoules. So this allows to do tomography. So this is a proof of concept. We initially did this, is that we put a tube below a 3 centimeter chicken breast. And I was amazed, we still can see a signal from this tube after 3 centimeter chicken breast. And you can see a delay, that's the delay of the sound. So this was, and we can see an image. This is after 5 millimeter chicken breast, but still that the image is very clear from this polyethylene tube. Well, this is the speed is slow. It takes 20 minutes to record the whole image, but we can do an array. And so now we actually, this is an unpublished image that we can put an array here. And this is the carotid artery from the peak. And you can see the ultrasound tells you the morphology, but now this tells you this is perivascular fat and the intravascular fat. And we have a spectrum, right? So each pixel we actually have a spectrum. This spectrum tells us this is a lipid. And we actually can analyze the composition of this lipid by this additional analysis.
What's the spectrum look like outside the?
It's different, yeah, it's different. So we actually can separate them. Yeah, that's right. So we can now scan the whole brain. So this is ultrasound image. So this is ex vivo. This is fresh brain. You take it and you scan. Then what do you see? You actually see the fibers. So this is the corpus callosum, and there are other. So basically, we can now map all the white matter in the brain by this method. Well, I hope that we can do in vivo soon. We have to thin the skull. The skull actually blocks the sound. But if we can do in vivo, then this can be used to study, for example, multiple sclerosis progression. Yeah, and not in human, but in small animals, that we can do the in vivo imaging of the myelin degradation and regeneration of these things. And there could be others to do this.
What resolution is that?
Resolution is 100 micron in this case. So the resolution is determined by the ultrasound. So in this case, we actually use a high frequency ultrasound. We use 20 megahertz. To go to human, we need to go to 5 or 10 megahertz, and that will further reduce the resolution to 200 micron. Well, I want to stop here, and I will show you what we do, right? So in my lab, we do three things. One is we develop imaging tools, basically label-free imaging tools. But we do a lot of basic biology. I enjoy that to discover new biology process. But meanwhile, we also build these tools to deliver these medical imaging tools. And this is based on these intrinsic molecular signals. So this is my group. So this shows a style we're doing that my group is very unique, is that we have students from physics, chemistry, biology, and engineering all together. So it's a very multidisciplinary team here. And we have funding from NIH and from a few foundations, DOD grants. And finally, that I want to show you that we also have a workshop and in July. So this is a more has a narrow topic. This is exclusive focus on spectroscopy and label imaging. But we have each year we have a high-profile list of speakers every year, and we also have a one-day training that show you this instrument, and also the how to analyze the spectral data. We have the software how we do the multivariate analysis on the second day. So you can contact me if you are interested. Well, thank you for your attention.
Question, please.
Yeah, please.
Like in SRS and CAS, you use femtochecking pulses. So, but width in the frequency domain will be more.
Exactly.
Yeah, but in frequency domain, the width will be around some wave number. So, how much you can resolve in this fingerprint region?
Yeah, that's a good question. So her question is that well, her question, this question, that's a good question, but it took us years to answer that question actually. So her question is that if the femtosecond is very broad, right, how can you resolve the fingerprint bands? Yeah, that's a very good question. And when I give a talk a few years ago, people exactly ask the same question. So initially, I want to do the femtosecond is actually it's a no-brainer actually because I want to promote the field, and the best way to promote the field is to do multimodal imaging, to use the strength of multiphoton fluorescence, and but then that loses the resolution. So now we solve that problem. So the beauty is that the femtosecond pulse contains all the spectral component to do spectroscopy. If there is a way to resolve those components, and that can be resolved through the pulse shaping method, because pulse shaping allow us to yeah, there are a lot of details, but you see all the components there, right? So if you have a way to use them, you actually can produce a spectrum at every pixel, and that creates a lot of capacity that a single color cannot be done. So that's
And in that case, you have resolved this 2 cholesterol peak, and you have shown the imaging using ELSA. So have you given something for training, like pure spectrum?
Yes. Training, you mean training, right? Yeah, yeah. That's a very again a very good question that we are also building. That so if you know the target, so if you know your goal, for example, if I know my goal is to I want to measure membrane, and I want to know if this membrane is on action potential, off membrane potential, for example. Let's give that example. I don't need to measure the whole spectrum. If I know the profile is different, I can build I can train or build a filter, right? And I can tell that whether this is tumor, non-tumor, yeah. So that is doable, yeah.
And one more question regarding this diffuse photon. So, like in SORS also, people do the same thing. Yeah. They are also reading the diffuse photons only.
Diffuse
in SOR, surface offset Raman spectroscopy.
What's the question?
So, like, what are the benefit of this SRS over that.
Over what?
Over SORS, the surface offset Raman spectroscopy.
You mean SIRS?
No, it's not SIRS. It's like they probe at one position and they detect at the other position.
Spatially offset Raman compared to SIS. Was yes, a spatially offset Raman is very nice because it can measure deeper tissue, right? And that's a very, very nice way to do it. SIS is the goal of SIS is to gain the speed back. And the advantage and disadvantage is obvious. SIS can be done at the room ambient light, for example, right? Because you measure laser, so that's a big advantage. In the surgical room, I don't think a surgeon want to turn off the light to do a Raman, go back and turn on the light to see what happened. That's the one thing, right? And if you open the tissue, maybe the depth is not very essential anymore. So, but again, these techniques all have their strengths and weaknesses. They all complementary to each other. Yeah.
So in your last comment, basically, obviously there's strength and weaknesses. So you showed this spectroscopy images like 30,000 frames per second, which is like really amazing. So what do you think? Where are the some of the weaknesses of this technique? Because if you can get this spectroscopy image like in tens of microsecond, so why use other techniques? Compared to fluorescence, for example, what I can see is that in fluorescence the background signal is like zero, but if you look at a specific target, you can tag it and you don't see anything else. I can see you can have some backgrounds. In general, some comments about this technique compared to other techniques out there.
Yeah. No, that's a good question. So actually that was in my yin and yang picture. So you gain the speed by narrow the bandwidth. So in this case, I record a 30,000 Raman spectrum, but I should tell you that that was not the whole window, right? It's actually, in this case, 200 wave number. The Raman is 3,000 wave number, and we can go broader. But if I go to 3,000 wave number, I will lose it. The reason is because then I will damage the sample, right? I have to use more photons. So SIs or CAS SIs are most useful if you know your target. And in many cases that's known because you know your goal is to distinguish cancer with non-cancer or to study a biological process. But if we know nothing, then the spontaneous Raman provides the best capacity to make a discovery. And I'll show you an example, right? We don't know anything, so we just take a spectrum and then found, oh, that's cholesterol, not a lipid. Yeah.
For tracking drugs, what's your opinion on using these endogenous signatures versus configuring and various drugs? Using a much better SMR.
Due to the drug. Oh, you mean endogenous, you mean labeling method?
Labeling drugs, since you're putting it in labels.
Yeah, so yeah, sure. That's if you use a CD bond, well, in labeling that you make the image easier to do, right? You can do single color because that's a CD bond. But in principle, if you can record a spectrum, you don't have to do it, right? You just do pattern recognition now, yeah. So so it depends, yeah. I think the advantage to me that this Raman probe, I think the value is that if someone can develop a Raman probe, the signal is much bigger than endogenous mode, then you win. But otherwise, you don't have to do it. Otherwise you can achieve the same goal almost with the label-free method. But again, we still have a big challenge, which is that no matter how you make, it's still much weaker than fluorescence. Even the one we made, the molecule we made, we achieved the detection sensitivity of 13 micromole. Probably that's better than others already, but 13 micromole is very for fluorescent people, they think that's too much. Yeah. We try to reach sub-micromole, but fluorescent have a single molecule already. Right? So, and that's a big problem because inside cells, most molecules, the concentration is at the nanomole. The drug, especially drug, right? People use drug as nanomole, treat the cancer cells, and that presents a big problem. Yeah.
Yeah, questions? Yeah.
What could be the maximum spectral range you could obtain through CARS? Because I have seen people are using like the lipid region only, the CH stretching mostly. I've not seen like people are doing in the fingerprint region.
What's the question of fingerprint?
The maximum spectral range in CARS you could obtain, like you can image in one shot.
Oh, you can as broad as you can. Actually, there are people doing multiplex CARS, like what's the name? Yeah, they can cover the whole range, very broad. Yeah, you can cover the whole range. They cover from 500 to 1800 to the whole finger broadband CARS. You can cover that. What's the name? The difficulty there is that they still take tens of milliseconds per pixel or tens of minutes per image, and then that go back to Raman because that speed can only study fixed tissue. And is that your question? What's